All righty. Well, thank you for having me. This is a new venue for me, definitely, and it'll be really tough to follow Dr. Brinkman, who was there in the PhD program at UBC. So, anyways, I'm going to talk at a little bit of a higher level, a lot less data than we've just seen for sure, but I think about an important strategic initiative at BC Children's Hospital Research Institute, or BCCHR. So the researchers at BCCHR are really a diverse faculty spanning multiple different UBC departments and faculties. About 250 total faculty, 1,000 or so researchers, with a large proportion, 70%, that are active in practice at BC Children's and Women's. And that partnership really between BC Children's and Women's and the Research Institute really powers this bench to bedside translational research. So precision health technology, this can be defined in several different ways. Here, largely thinking about the omics, the genome, transcriptome, epigenome, proteome, metabolome, microbiome, fMRI, flow cytometry for sure. All of these different technologies, they really provide insights into health and disease at the cellular and molecular level, but they come with lots of challenges when it comes to applying them on patient-centered research projects. So if we think about the patients, one of the most vulnerable groups at BC Children's are really these patients on the diagnostic odyssey who have reached the end of the line for the clinical standard of care, and it's time for them to transition into the research space to access these new emerging technologies. So when we think about the current state, or even the previous state before we really got going, these different isolated silos of expertise for different pieces, whether it's proteomics or single-cell transcriptomics, the expertise existed, but there was not a very streamlined way for patients to access them. Furthermore, there's lots of redundancies when it comes to each one of these silos or islands, as you see in this map with this diagnostic odyssey of puzzle pieces. And those include redundancy around computing infrastructure. We had several different small high-performance computing systems, sometimes assigned to different cores at the Institute. Ethics approval can be a challenge, especially for clinicians who are bringing patients the need to access maybe one of these technologies, but getting ethics approval can be challenging. And then you've got this real large burden of training. And so every time one of these technologies emerges, oftentimes it's researchers, sometimes bench researchers, who are told, maybe by their PI, go learn how to do it. And that can be quite challenging and lead to, really problems that we see multiple different researchers having. And so with the Precision Health Initiative, we wanted to turn this kind of puzzle piece odyssey into something that's more streamlined so that any patient-facing research project at the hospital could then access any of these technologies if they needed to. And we really would be focusing on kind of two different components that we would help provide to improve that connectivity. One is coordinating computational solutions for the researchers. Another is about training. And so how can we share in training, whether it's for high performance computing or specific to one of these data modalities, in a way that's shared across the Institute and the larger UBC ecosystem. We also provide, through something called the Rare Disease Discovery Hub, a shared PHSA and UBC approved ethics consent protocol. So if you're a clinician bringing a patient who needs to access one of these pieces, we have a mechanism to enroll them into the program and then partner with the clinician to essentially find answers for their patients. Now, this will lead to, obviously, diagnoses and potentially treatments, improving the collaboration across our institutes, publications, highly trained personnel, all these great things that really help drive the Institute to the next level. Now, our three focuses in early implementation include applied precision health research in the clinical setting, training and education in precision health technologies, and finding solutions for computational solutions for researchers. So the first I'm going to focus on is the applied precision health research. So our focus initially is on rare genetic diseases. Now, the burden of rare genetic diseases, individually, they are rare, but collectively, they actually have a large burden both on our hospital system and on our population here in Canada. About 1 in 25 children are born with the rare genetic disease. They are increasingly taking up about a third of the pediatric hospital beds. And unfortunately, a quarter of these children won't make it past their 5th birthday. We're also falling far short of diagnosing these patients. In fact, roughly 50% of cases remain undiagnosed even after accessing things like exome sequencing or panel sequencing. And then a problem that we haven't ventured into yet, but definitely would benefit from having connectivity across our campus, is that 90% of diagnosed cases still will lack a treatment. That number is more likely even higher. So why is it such a challenge? Well, if we think about the scale of the genome, we're talking about 3 billion base pairs, more or less 22,000 protein coding genes, about 5,000 known disease-causing genes. So finding that single nucleotide variant somewhere in those 3 billion base pairs can be a lot like finding that needle in the haystack. Now, with whole genome sequencing, we can more quickly move from a sample in a patient into a likely diagnosis by finding a pathogenic variant. So by starting with DNA, using an Illumina NovaSeq down at the Genome Sciences Center, we get whole genome sequencing reads. These are just short fragments of DNA. We then can align those to a reference genome, figure out the places where there are mismatches or representing a variant between the sample and the reference. And then we take those genomic variants, somewhere around 3 to 5 million per person, and we annotate them and filter them. First, looking at things like, is this variant already known to be clinically relevant? Is the gene known to cause a disease that matches what the patient is, the symptoms the patient is showing? We also look at population databases. These have really grown rapidly in size, especially with the UK Biobank and the NOMAD data set, such that we can now get a decent idea of, is this variant common in the population, or is it really as rare as this 1 in a million disease that we are investigating in the patient? We have a number of in silico predictors. So this is to say, do we predict this variant will have a pathogenic impact? One of which has been very powerful recently is called Splice AI. And this is a method which can predict whether or not a variant will have an impact on the splicing, whether it's the acceptor gain or the donor loss, that could lead to aberrant splicing, poison exons, frame shift variants, a couple of which I'm going to talk about in just a second. And then lastly, inheritance. So is the variant following an inheritance pattern, segregating in the family, segregating with the phenotype? And if it's a compound heterozygous variant, are the two in trans? So we go from about 3 million down to a handful that we classify into categories. So many of you will have likely seen these categories from the American College of Medical Genetics: benign, likely pathogenic, pathogenic, and then this large category in the middle where most results end up: variant of uncertain significance, VUS, VUS. People call this a lot of different things, but it's a real big headache. Now, the reality is that these VUSs can often be moved into either benign or pathogenic state by applying additional assays. That could mean inheritance testing for segregation, cellular or animal modeling, especially in cases where there's a likely developmental defect, or even molecular and biochemical profiling, things like transcriptome sequencing or investigating the metabolome. And this is where our precision health toolkit comes in, that we can reach into this toolbox once the clinical standard of care has been reached, and we can apply these technologies, whether it's segregating a variant, collaborating with the researcher at the hospital to do cellular animal modeling, or even just applying something like transcriptome sequencing to resolve with RNA-seq. And so this patient story really starts about 12 years ago, where there was a man who came to VGH and was seen by Dr. Burkle. And Dr. Burkle recognized that he had these very characteristic melanotic macules on the lips, which indicate Peutz-Jeghers syndrome, which is a defect in the gene STK11. Now, the variant that was identified was a VUS, was 6 base pairs into the intron, and thus not classifiable as pathogenic. So nearly a decade goes on, and this man's son then starts to develop the same features. Now that there's a father and son, the variant is genotyped in the son, and it turns out he also shares that same intronic variant. Now, 5 base pairs into the intron is not necessarily what we would consider for the critical splicing region, but when we investigate what that variant's actually going to do, is it's creating a new A to G acceptor site. So this variant is at this position 6 base pairs in, goes from an A to a G, and with a new AG acceptor site, that means that their splicing machinery can now recognize this site, especially with the polypyrimidine tract just upstream, as the primary splice site. So this was the hypothesis. If we apply RNA-seq, we'll see evidence of this. And just with short read RNA-seq, followed with Sanger sequencing of the cDNA, just because we still need to confirm things with Sanger to make certain people happy, we found that indeed there is a 5 base pair inclusion of that final mRNA. That 5 base pair inclusion ends up leading to a frame shift and a premature stop codon. So with this diagnosis, really the relief on the father's face after a decade of worrying was striking, as Dr. Burkle recalls. And with the diagnosis, what's important about this disease is that there's a very stringent management plan that includes very frequent essentially screening for polyps in the GI tract, because that's one of the main cancer predisposing features of this syndrome. We published this paper in the American Journal of Medical Genetics if you're interested to read more, but this was really not a one-off. In the past 8 months, we've applied RNA-seq and resolved 4 out of 4 additional cases. These, unlike cancer predisposition, were more intellectual disability and developmental disorders, but nonetheless, with enough gene expression in the blood, a simple blood sample followed by RNA-seq was enough to show that indeed we can move a variant of uncertain significance into that pathogenic label, which leads to, with a diagnosis, these families can access things like management, certain patient resource groups, and really have a better understanding now that they have a diagnosis for their disease. Within the Precision Health Initiative, we're ramping up to resolve about 50 cases per year using transcriptomics, as well as applying things like Oxford Nanopore long-read sequencing when we might not even have a variant discovered from the classic exome that these patients have received. So beyond the applied precision health research, there's a big emphasis in training and education. Now, we have 2 efforts, one more on the clinical side and one more on the research side. So from a clinical standpoint, Dr. Stuart Turvey, who's the lead of the Precision Health Initiative, you'll see his face on the last slide here, has a vision that he should be able to walk down the hall of the hospital, show a variant description to any clinician, and they should be able to understand what is that variant, what does the amino acid change mean, when it's an intronic variant or a variant of uncertain significance, what does that mean? Currently, he says that about 30 to 40% of clinicians could actually interpret that variant report. But as these are being distributed all across the hospital setting, no longer just confined to medical genetics, it really becomes something that needs to be trained frequently for active clinicians. And so to that end, we have been focusing on some clinical efforts, including hands-on workshops and lectures, primarily to the pediatric subspecialties, the allergy and clinical immunology fellows, and even the genetics residency training program. We also teach a module to the genetic counseling program, as we see that a lot of this kind of bioinformatic analysis really can be shifted into the genetic counseling domain, such that genetic counselors who are seeing patients, they should also be able to interpret variants and have a good understanding of how you go from a blood draw to a variant on a report, even if that report's coming from somewhere like a commercial lab in the US. And then lastly, the research-focused efforts. So here, working with the bioinformatics program to try and engage students in this space of undiagnosed diseases. I think there's a lot of opportunity there, especially when it comes to the nanopore space, as well as one of our latest achievements, which was the Precision Health Bootcamp. I think also the reason why I'm here, which was an effort that we had in August of this year, that really was a collaboration between Precision Health Initiative, UBC's Advanced Research Computing, and the Trainee Omics group at BC Children's. Now, all in all, this was a workshop that had about 90 attendees from across the UBC and affiliated institutions. It ran for 3 weeks, 2 per day, and I will never do that again. We learned some lessons there, but I wanted everyone to be able to attend every session if they wanted to, and my God, we had about 10 people who did. All in, we had core sessions, which were more about how do you actually use these systems, teaching the command line, high performance computing, the scheduler, as well as a little bit more advanced getting into maintaining your own software stack because you really can't trust anyone else. And then getting into the applied sessions where we had different researchers, grad students, as well as a couple of assistant professors teach applied sessions on things like NextFlow, RNA-seq, the single cell, DNA methylation, GWAS, multiomics, among others. This content does remain available online. We've got recordings of all of the videos that we had in the session, and it has been accessed asynchronously by a few different people. So if you're interested in pointing towards this or perhaps in participating in future years, please get in touch. And then the last piece here really is about coordinating and finding computational solutions for researchers. Now, in my PhD, I had to access many different systems, although we are now moving to a space where those systems, some of them are retiring, some of them are not adequate for sensitive patient data. And we're moving away from this model where your PI has a big computer somewhere, and that's enough to handle the data that you've got. So one of our efforts really has been in centralizing and tearing down everyone's individual HPC and integrating them into a large cluster, gaining some additional investment, and we're supporting on our local compute cluster at BC Children's about 80 different researchers. It's also been important to engage with UBC Advanced Research Computing towards the effect where we can really get a better idea of what are the needs of our researchers and how are they being met by the compute providers. One of the big gains there has been in coordinating with PHSA projects like Care for Rare, the Causes Project, that we've been able to migrate data, which we used to, by our ethics protocol mandate store in-house. We can now move that to the UBC platform, which is much larger and honestly much more suitable for data of that magnitude. And then lastly, we've been waiting for the Digital Research Alliance of Canada. Maybe others of you have as well. We're not holding our breath, but when it does come available and when that is suitable for this sort of identifiable genomic patient-focused data, that is something that we will explore, and then we'll also provide training on how our researchers can actually utilize those systems. And with that, I will conclude. Here's a smiling photo of Dr. Stuart Turvey. He is the lead of the initiative. He's also the Aubrey J. Tingle Professor of Pediatric Immunology at UBC and a Canada Research Chair in Precision Health. Russell Bonaguro, who is the project manager of the initiative. And we really are open to collaborate and integrate across the UBC Faculty of Medicine. Please get in touch if you're interested in either the training, the Rare Disease Discovery Hub patient-focused efforts, or if you just want to chat about precision health.
Great, thanks very much. Are there any questions from the room here? Yeah. I think someone else's mic is up. Do you want to maybe go ahead first? And then
Hi,
we'll go to
thanks. That was very interesting. When you discover about one or like you resolve one VUS per week, how do you share that knowledge with the community?
Yes, so in a couple different ways. One, it actually, in order to apply that RNA-seq and assert a diagnosis, you actually have to publish that case. And so it's this interesting piece that even if we have the data in hand, we can show that there is an effect. The clinician is convinced. In order to provide that diagnosis, it actually has to go into the literature. So to date, we've been doing this with things like case reports. We're also having a big push for submitting the variants that we do assert as pathogenic into open source databases like ClinVar. I think this is something that, really should be done for every single diagnosis given. There's overhead in submitting these things, but once that goes in, now if the same variant is discovered elsewhere, somewhere else in the world, and this does happen, now they'll be able to see, a link to the publication. We'll see it in ClinVar, and then that might provide a diagnosis without the need for that extra assay.
Okay, and up here.
You mentioned some examples of the variants with unknown significance for some of where you sort of associated them with the phenotypes. Did those individuals only have that single VUS? Because I work in sudden cardiac death as well, where individual patient can so many VUSs, and then it's like, how do you attribute a single one to the phenotype?
Yeah, so the approach to resolving VUSs can be challenging. I think there's a couple aspects there. One, there tends to be in that VUS category even further labeled. So you can have a VUS that's more cold or one that's more warm. And that differentiation between a cold VUS and a warm VUS might be things like an in silico predictor score, how it's segregating in the family. So in the end of the day, there are more ways to rank different VUSs, but there's also been some really interesting results recently from the Undiagnosed Disease Program, which has shown that if you're receiving panel sequencing, you actually have more VUSs than if you were to get a whole exome. And part of that is because when you're using that panel, you're kind of forcing yourself into being convinced that there is something there. So I think as we step back and go more genome wide, we'll have a better idea. But at least in the cases we've seen before, this has really been a, lead candidate VUS. But if you apply RNA-seq and that doesn't end up panning out, you still have expression across the entire genome, at least what would be expressed in blood, to look at other signatures. And so we've got a couple different tools that we're using, which we can plug in an RNA-seq data set, and it will tell us what are the outlier expression genes, are there outlier splicing junctions that are not seen in the population. So there's extra things you can do with the data in hand, but you're right, chasing VUSs can be a very challenging endeavor, especially in something like that cardiac space.
What percentage of the requests do you actually carry out the analysis on?
That's a great question. So at least in the Rare Disease Discovery Hub, the way that it works is there is an expert panel of clinicians and researchers where every case will be presented, and then there'll be a decision as to whether or not it goes through. And so I would say, in general, the uptake from the Rare Disease Discovery Hub is that probably 70% of the cases would go through to some next step. In some cases, things get presented and it's just not feasible or it's too high risk. Like, for example, if it's a splicing variant and something only expressed in the brain, that's going to be really hard to, justify going after. But that being said, certain aspects of resolving a VUS can be very low burden. And so, for example, segregating in the family, that is not even covered by MSP in certain cases. We can do that with a very low overhead Sanger sequencing, and that can then justify the next step.
And at the other end of the process, what does your report look like?
Yeah, so we write research reports, essentially including the investigation of the omics data. This is just a PDF report that would then go back to the clinician. And then in some cases, they can take that to get an additional clinical test, like, for example, a clinical Sanger. Sometimes that's needed for the patient chart, but we are not producing clinical reports from our process. It's still a research enterprise.
Thank you.
All right. If there aren't any other questions from the audience here, I don't think we have any online. Thank you very much.
Thank you.